every single day, without you even realizing it, your body is a literal battleground. I mean, your physical barriers, like your skin and mucous membranes, are just continuously bombarded by this invisible world of microorganisms. But what happens when those walls are breached? Well, in this explainer, we're zooming way in to look closely at the microscopic war raging inside you right now. We're going to focus specifically on how your immune system's frontline soldiers execute their targets. So let's dive right in and set the stage for an epic cellular battle. It's a kind of terrifying thought, isn't it? Just a splinter in your finger, a scrape on your knee, or breathing in a pathogen. When bacteria successfully slip past your physical defenses, they enter this warm, nutrient-rich environment where they want nothing more than to just multiply. But waiting for them right beneath the surface is a highly specialized, absolutely ruthless defense force. Honestly, if these early responders were to fail, a minor scrape could literally be lethal. So how exactly do they neutralize the threat? Here is our battle plan for this explainer. First, we'll meet the cellular defenders. Second, the hunt and capture. Third, the execution mechanisms. Fourth, how bacteria fight back. And finally, winning the microscopic war. Section 1. Meet the cellular defenders, the cellular frontline. Let's start by getting to know the frontline soldiers of your innate immune system. These are the units deployed the literal second an invasion is detected. The strategy these defenders use is called phagocytosis, and the literal translation of that word, cell eating. It is absolutely fundamental to your innate immunity. You see, these specialized cells don't just poison invaders from afar. They physically recognize them, engulf them whole, and digest them. It's an incredibly aggressive and effective way to clear out invading microorganisms, dead cells, and cellular debris. So who are these professional cell eaters? Well, we've got three main players. First up, the neutrophils. Think of these guys as your rapid response SWAT team. They circulate in your blood, rapidly migrate to infected tissues during acute inflammation, kill microbes aggressively, and then they die shortly after. They have a really short lifespan. Next, we have the macrophages. These are the long-lived sentinels residing in your tissues. They are the heavy infantry. Not only do they engulf and destroy pathogens, but they actually coordinate the wider immune defense by sending out chemical signals. And finally, the monocytes. These are circulating reinforcements in your blood that, when called in to infected tissues, actually differentiate and transform into macrophages to join the fight. Section 2. The hunt and capture. Tracking the prey. We know who the hunters are, but bacteria don't just sit still and wait to be eaten, right? Let's shift our focus to the tactical phases of the attack. This brings us to a highly coordinated 4-step capture sequence. Step 1 is chemotaxis. When tissue is injured or infected, chemical signals, like the complement fragment C5a or bacterial products, act just like a scent trail. The phagocytes follow this chemical scent directly to the site of the infection. Step 2 is adhesion. Once the hunter reaches the prey, it has to grab hold. Receptors on the phagocyte establish firm contact with the microbe. Then, step 3 is engulfment. The phagocyte extends its plasma membrane, literally reaching out with these arm-like structures called pseudopods to completely surround the bacteria. And step 4 is phagosome formation. The pseudopods fuse together, trapping the microbe inside a specialized, sealed membrane vesicle within the cell, known as a phagosome. But, sometimes bacteria are slippery and really hard to grab. So to make adhesion incredibly strong and engulfment highly efficient, your immune system essentially paints the target with molecules called opsonins. The most important ones here are IgG antibodies and a complement protein called C3b. Think of opsonins like Velcro or maybe a tracking beacon. Once a bacterium is coated in C3b or IgG, the phagocytes receptors lock on with incredible strength. The whole process transforms from a difficult struggle into a highly efficient capture. Section 3. The execution mechanisms. Inside the kill room. Okay, the target is captured and trapped inside the cell, but it isn't dead yet. In fact, inside that newly formed phagosome, the bacteria is perfectly alive. Let's enter the climax of the attack, the kill room. The initial phagosome isn't actually deadly. To become lethal, it has to mature. The cell pumps acid into the vesicle, dropping the pH drastically. Then, this highly acidic phagosome fuses with intracellular sacs called lysosomes. Together, they create the phagolysosome, an inescapable kill room. This fusion unleashes a touring of hydrolytic enzymes, antimicrobial peptides, and a highly toxic environment designed to utterly dismantle the engulfed microorganism. So the absolutely crucial point here is how the actual execution happens within this chamber.
The phagocyte uses a brilliant dual-threat system. On one side, we have oxygen-dependent killing. During phagocytosis, the cell rapidly consumes oxygen in an event called the respiratory burst. An enzyme complex called NAPDH oxidase takes that oxygen and converts it into superoxide, which then cascades into hydrogen peroxide and other highly reactive oxygen species, or ROS. In neutrophils, an enzyme called myeloperoxidase even mixes hydrogen peroxide with chloride to create a powerful bleach-like oxidant. This literally burns the bacteria chemically, damaging its proteins, lipids, and DNA. On the flip side, we have oxygen-independent killing. Because even without oxygen, the phagocyte is lethal. It unleashes lysosomes that just chop up the bacterial cell wall. It deploys defensins, which are these peptides that punch holes in the microbe's membrane. And it uses lactoferrin, a really clever protein that steals away iron, literally starving the bacteria of the vital nutrients it needs to survive. Through this violent oxidation and chemical degradation, the bacteria is utterly destroyed.
Section 4. How bacteria fight back. Microbial countermeasures.
It sounds like an unbeatable system, right? Well, nature is all about balance and adaptation, and engulfment does not always guarantee a kill. Pathogens have been evolving alongside us for millions of years, and they've developed some absolutely stunning countermeasures. Some bacteria use a physical shield. They grow this thick, sugary capsule that hides their surface molecules, making it incredibly hard for the phagocytes to recognize or grab them. Others team up and use biofilm formation, encasing themselves in a dense matrix that phagocytes simply cannot penetrate. But what if they do get swallowed? Well, some cunning bacteria actively trigger the inhibition of phagosome maturation. They chemically block the lysosomes from fusing with the phagosome, meaning the kill room is just never activated. And perhaps the most dramatic strategy of all, escape into the cytoplasm. Before the lethal enzymes can be unleashed, the bacteria breaks right out of the vesicle and swims freely in the fluid of the host cell, where it is completely safe from destruction.
Let's see how this plays out in the real world with some absolute masters of survival. Take Mycobacterium, the culprit behind tuberculosis in humans and various animals. Once swallowed by a macrophage, it actively prevents the Phagolysosome from forming. The macrophage essentially becomes a protective bunker where the bacteria happily persists and causes chronic infection. Then there's Listeria. It uses that dramatic escape route we just talked about. It breaks out of the phagosome completely, enters the host cell's cytosome, multiplies, and even hijacks the cell's own internal machinery to launch itself into neighboring cells. And Salmonella? It actually modifies the internal vesicle it's trapped in to create a cozy, safe little niche to survive and replicate after being engulfed. Successful pathogens don't just avoid phagocytosis. Some have literally evolved to use the very cells meant to destroy them as a protective home. Section 5. Winning the microscopic war. Long-term immunity. This constant back and forth between host and pathogen really dictates our health. But what happens when the phagocyte actually wins and the bacteria is successfully destroyed? Well, it isn't just about cleaning up the mess. It's about preparing for the future. After a successful kill, the phagocyte degrades the microbial debris. But it doesn't just throw all the trash away. Macrophages take specific fragments, peptides, from the dead bacteria and display them on their surface using specialized proteins called MHC class II molecules. This is called antigen presentation, or passing the intel. The macrophage travels to the lymph nodes and shows these fragments to T cells. This is the crucial moment where the rapid innate immune system hands over the specific intel to the highly targeted adaptive immune system, granting you long-term immunity against that specific invader. Looking at this incredible biological machinery, there's a quote from our source material that just perfectly summarizes this entire explainer. Phagocytosis is a battle between the killing machinery of the host and the survival mechanisms of the pathogen. It is never, ever a passive event. It is an active, violent, and strategic microscopic war happening within your tissues every hour of every single day. We've seen the weapons, the tracking beacons, the acid chambers, the reactive oxygen bleach, and of course, the bacterial armor and escape hatches. It is a continuous, evolutionary arms race that has been raging for millennia. As our cells develop better ways to hunt, pathogens evolve better ways to hide. So, I'll leave you with this to ponder. In an arms race where both host and pathogen are constantly evolving new ways to outsmart each other, who do you think will ultimately gain the upper hand? Thank you so much for exploring this microscopic battlefield with me. Keep learning, and I'll catch you in the next explainer.